Hi everyone. So we are starting a new unit today on cell division and DNA structure. So we are going to be splitting this into a couple different sections based off of first DNA structure and then cell division. Just trying to get to the big questions over here. So some questions we're going to be trying to answer over the course of this unit are: why do cells divide? Like, why do we need to make new cells? How do cells divide? What does that process look like? And then why is DNA structure important for cell division? So why is it important to understand the structure of DNA, how it's put together, so that we can better understand its function in terms of cell division? Because remember, every cell needs a full copy of DNA. So we're going to be starting specifically with DNA structure. At this point, I believe most of you should be at least familiar with DNA. We should know that it's sort of like the genetic code of life. It codes for all of our traits and characteristics. It is found in the nucleus of the cell, and every single cell in your body has a full copy of DNA. So keep in mind, your liver cells use some genes in your DNA and not others, while your brain cells use other genes and characteristics in the DNA, but not others. But both the liver cell and the brain cell will have a full copy of DNA, including even the genes that they don't use. So keep in mind, out of all of the billions and trillions of cells that make up the human body, every single one of those billions and trillions of cells will have a full, complete copy of DNA. So, like I said, we should be relatively familiar with DNA in terms that DNA stands for deoxyribonucleic acid. You never have to say that out loud. You never have to actually spell that correctly. But DNA, so D is for deoxyribo, the N is for nucleic, and the A is for acid. So remember, nucleic acid is one of our 4 macromolecules. So now that we've gone through the digestive system and we've talked about proteins, carbohydrates, and lipids, now we're looking a little bit closer at DNA as our 4th macromolecule. So DNA, like we've said, is made up of nucleotides, so the base unit of a nucleic acid. And there's 4 different types of nucleotides. We will be going into this in more detail on a future slide. It is in what we call a double helix shape. So double helix is sort of like a twisting, winding staircase type of a shape. Double is because there are 2 strands of DNA. So if you look at this image right here, this is showing you that double helix shape. So this is definitely twisting around. It sort of looks like a twisty staircase. And so one strand of DNA would be this one side of the DNA over here, and then the second strand of the DNA would be this side of the DNA right here. So that's where we're getting the double-stranded or double helix shape. And like I've said, DNA stores our genetic information. It's sort of like our blueprint to create us the way that we are. Now, just a little background on DNA. So, like I've said, deoxyribonucleic acid is what DNA stands for. We know it's made up of 4 different nucleotides in that double helix shape, and it stores genetic information. Now, to give you an idea of just how much DNA is inside every single one of your cells, if you take the DNA out of the nucleus, you stretch it all the way out, you can wrap it around the nucleus, so it's the diameter of the nucleus, 100,000 times. Okay, so we're gonna get to this metaphor down here in a second, but I'll come back to that. So if you take the DNA out of a cell and you stretch it all the way out, it's going to measure about 2 meters or 6 feet. So keep in mind, 6 feet is a relatively tall person. So that means even if you are a shorter person, like a 5-foot person, if we take the DNA out of your cell and stretch it out, it's still going to be about 6 feet long. And we need to take this 6 feet of DNA and shove it into this itty bitty tiny little nucleus inside of your cell. So the comparison I'm going to give to you is if we take 113 miles of string and stuff it into a basketball, that's what it's like to stuff the 6 feet of DNA into the nucleus. Now, 113 miles of string. This is if you were to like drive a car to Hartford, Connecticut and back to Worcester. So from Worcester to Hartford, Connecticut, back to Worcester, round trip, you let a little tail of string flow behind your car as you drive, and you take that string. It's 113 miles, so round trip 113 miles of string, shove it into a basketball. That's what it's like trying to shove your DNA into the nucleus of your cell. So the DNA does have a very specific structure in terms of packing itself into the nucleus of your cell, and we're going to be going over that. This is a diagram of what it kind of looks like right here. But like I said, it's a massive amount of stuff shoved into an itty bitty tiny little space. So hopefully the question you're asking yourself is, how does all that DNA fit inside the nucleus? Basically, the DNA is going to coil itself very tightly around a protein called histones. These histones kind of look like hockey pucks, and it just is an organizational method for your DNA so that it can, one, package itself into this tiny little space, but two, help keep it organized so we know where the different genes are located in the DNA. So when your DNA does actually package itself down into its most condensed form, you're going to get either a chromatid or a chromosome, depending on what's going on. So chromatid is supercoiled DNA that copies itself before cell division. Chromosome are two chromatids held together by a centromere. So this picture right here is a chromosome. Hopefully it looks semi-familiar. I think most of us have seen like an image of a chromosome before. So this entire thing is a chromosome. However, this side right here would be a single chromatid, and then this side over here would be your second chromatid. So to make a chromosome, you need two identical chromatids. So this is identical to this right here. And these two chromatids are held together by what is called a centromere right here. And that's how we get that sort of like X shape of the chromosome. It's not really an X, it's just two like things held together. But the idea is, like we've said, we're going to coil the DNA up until it reaches this highly condensed state. So this DNA here, this is our double helix. This is, kind of how we're used to seeing the DNA. It then will wrap itself around the histones, those proteins that look like hockey pucks. So this is a histone, this is a histone, histone, histone. It wraps itself around, and then it's sort of going to like wrap itself around itself again and again and again until we get that chromosome or chromatid shape that we are looking for. So this is a video right here that will show how DNA is able to package itself so you can get a better visual representation of how this DNA packing actually happens.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA
right
molecule.
Here. This is the histone we were talking about. So double helix,
the combined tight loop
histone.
of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes.
So these are actual chromosomes going through cell division. We again are going to get into this in more detail later this unit, but all of these little bumps and lumps are the chromosomes. So hopefully that gave you a good visual representation of what this DNA packing actually looks like to create chromosomes. Let me just make this bigger again. I believe in you, computer. Thank you. So for humans will have 46 total chromosomes or 23 identical pairs of chromosomes. So keep in mind when you are conceived, mom is going to give 23 chromosomes, dad is going to give 23 chromosomes, which means that 23 plus 23 equals 46, but each parent gives one of a pair. So maybe this this is a chromatid, but maybe this chromatid is from mom and this chromatid is from dad. They both have the same genes on them. So maybe we have like eye color, height, whether you have like free or attached earlobes, the ability to roll your tongue or not. They will have the same genes on them. They might not have the same copy of that gene. So what I mean by that is if eye color is located right here on the chromatid, maybe dad gave you a blue-eyed color gene and mom gave you a brown-eyed color gene. They both have the eye color gene in that location, but one might be for blue, one might be for brown. They could be the same, they could be different. Now, out of your 46 chromosomes, or out of your 23 pairs of chromosomes, there are 22 pairs, or 44 chromosomes, that we call autosomes. So auto, self, some, we're talking about the DNA. So these autosomes, these chromosomes that are labeled as autosomes, so all the pairs up until pair 22 right here. So here's pair 1, 2, 3, 4, etc., down to 22. These all code for, I'm going to call them your usual traits. So like I was saying, eye color, hair texture, free or attached earlobes, the shape of your nose, whether or not you need glasses, how tall you're going to be, your skin color, all of these features about you. You have one pair of chromosomes called your sex chromosomes. That's what we're looking at right here. So in order to make a biological female, you need to have 2 copies of an X chromosome. In order to have a biological male, you're going to have 1 copy of an X chromosome and 1 copy of a Y chromosome. So to repeat that, biological females, 2 X chromosomes, biological males, 1 X, 1 Y chromosome. So looking at this image right here, this is called a karyotype. When we like display someone's these are chromatids. When we display all of someone's chromatids and we match them up like this, this is called a karyotype. So according to this karyotype, we are looking at the DNA of a biological male because there's 1 X chromatid and 1 Y chromatid. All right, so now let's get into the structure of DNA. How do we make this double helix? So the basic structure of DNA is made out of nucleotides. These are the nucleic acids that we've been talking about for macromolecules. So each nucleotide is made out of a phosphate group, a sugar group, and a nitrogenous base. That's what you're seeing in this image right here. So this would be one nucleotide. We have our phosphate, we have our sugar, and we have our nitrogenous base. For DNA, there are 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. They're usually called by the first letter of their name, so A, T, C, and G. You might have heard that before. And then the nucleotides are going to pair together to form the double helix shape. So a nucleotide that has an A for its nitrogenous base will always pair with a nucleotide that has a T for its nitrogenous base. And then a nucleotide with a nitrogenous base C will always pair with a nucleotide with a nitrogenous base G. So A and T pair together, C and G pair together. I remember which ones will pair together based off of how you draw the letter. So when you draw an A or a T, like right here, they're both drawn with straight lines. That's why they pair together. Scientifically, that's not why, but just to help you remember. So A and T pair together because they're drawn with straight lines. C and G will pair together because they're both drawn with curved lines. So that's what you're seeing in this image right here. So this is a nucleotide, so PSA, that's one nucleotide with the nitrogenous base of adenine with A. We have PSG, so another nucleotide. This one has guanine as its nitrogenous base, etc. And then you'll also notice here we have a nitrogenous base A pairing with a nitrogenous base T over here. G with C, T with A with T, C with G, G with C. So the idea is if we only had this strand of DNA, we would still be able to figure out what the second strand would be because of this base pair rule, because A will always pair with T, G will always pair with C, and vice versa. So this is just another diagram to show what DNA looks like. Looking at this image right here, it kind of looks like a ladder. So the sides of the ladder are known as a sugar phosphate backbone, and then the rungs of the ladder are where the nitrogenous bases are located. And then we take our ladder, we twist it up into the double helix shape, and this is just another representation of what it might look like on the atomic level. All right, so we have already kind of talked about the DNA double helix. It's double-stranded. There's 2 strands of DNA. So we're going to call one a template strand and one a complementary strand. So sort of like a template, like if you go to a photocopier and you want to make copies of something, you bring your template paper and that's what you copy off of. So your template is your original copy of something. In this case, the complementary strand, it's not just like saying nice things about the template strand. It's not giving compliments. But complementary just means that they pair together. So if you think about this in a fashion sense, not that I'm very fashionable, but you might say like one color is complementary to another color, or you might say like blue complements your skin tone. It pairs together well. So we have our template strand, which is our original copy, and then we have our complementary strand. So on the template strand, we can watch the A's, T's, C's, and G's, and we're able to create a complementary strand based off of the template strand because we know A pairs with T, T pairs with A, C pairs with G, G pairs with C. So like I said, that's what the template strand is, and that's what the complementary strand is. Now, this image right here does show DNA replication, which we will be getting into in the next video. So I'm not going to go into detail right here, except to show you that this blue strand of DNA would be considered your template strand, and the yellow strand of DNA is going to be considered your complementary strand. So we'll say that A is complementary with T, the puzzle pieces fit together, and C is complementary, or G is complementary with C, those puzzle pieces fit together. So at this point, make sure you have your vocab notebook open, you're going through all the different words. Make sure you're pausing the video, rewatching the video if you need to, but we do have a bunch of vocab words in this presentation, so please check in and make sure you complete those slides in your vocab notebook.